Thank you very much, Rob. That's very nice data and presentation. So in my presentation, I want to show you a couple of channel validation data on the next generation of the automated patch clamp system, IonFlux system. So the purpose of this study was twofold. One is to support the CiPA HASI high throughput screening efforts across different sites on different platforms, generate validation data. The second fold is that we want to test the feasibility of using IonFlux HT system in screening of the voltage-gated ion channel compounds, because we know that this platform is very good at the ligand-gated ion channel compound screening, but we want to test the feasibility of whether we can use this for voltage-gated ion channels. Before I go into the details of the data, I want to spend a couple of slides to briefly introduce you what we do, who we are, and what we do. Eurofins Scientific is a global company, actually a leading CRO serving mainly three different industries: the food test, the biopharmaceutical test, and environmental test. And we are leading, actually, #1 service providers in most of the fields in these three different industries. And now we're getting into the diagnostic industry too. We are established in 1987 as Eurofins Scientific, and now, as of the year of 2015, we have around 225 labs in 39 countries. We have roughly 23,000 full-time employees, and we have over 150,000 analytical methods available for the customer. The subsidiary or the business units that the ion channel services team was in is actually Eurofins Pharma Discovery Services. And this business unit was created by the integration and acquisition of three different legacy companies. In 2012, the Eurofins acquired the legacy Pan Labs. In 2013, the legacy Syrup, and combined them into Eurofins Syrup Pan Labs in 2013-2014. In the year of 2014, the Eurofins acquired the legacy Merck Millipore or EMD Millipore Discovery and Development Solution business unit, and take part of that and then integrate that into the business units that we're in now, which is called Eurofins Pharma Discovery Services. And here shows the global footprint and the map of the Eurofins Pharma Discovery Services. We have a site in St. Charles, Missouri, which is the ion channel service team is located in. And we have a site, at the site here, we also have the oncopanel services, the immunosignaling services, and as well as the amitoxin services. And we have a site in Dundee, Scotland, which is a home for the kinase profiler services, and we have a site in Poitiers, France, which is a legacy Syrup site. It's a home for a lot of in vitro assays as well as some cellular assays there too. And we also have a site in Taipei, Taiwan, which is a legacy Pan Lab site. They specialize in a lot of in vivo pharmacology tests as well as some in vitro assays as well. So like I said, by combination of all these different legacy services units that make us a full service provider that can cover your needs from the very beginning of your target identification and validation until your compounds goes into the clinical trials. And I want to give you a brief introduction about CiPA before I go into the actual data. So late 1990s and early 2000s, we have a lot of compound withdrawal from the market due to the torsade risk. And the scientists in the pharmaceutical industry and academia actually figured out this is largely due to the hERG blockage or hERG inhibition in the cardiac tissue. And hERG refers to a human interglobal-related gene channel, and then the current is referred as ITR, and channel also refers as QV11.1. So we know that the blockage of hERG can lead to a cardiac action potential duration prolongation and EADs, and which can lead to the QT prolongation and eventually proarrhythmic risk or torsade. So all these findings leads to the implementation of the ICH S7B and E14 guidelines early in the 2000s. And this hERG-centric strategy that we're using is very effective and somewhat successful because the evidences are since the implementation of these guidelines from early 2000, there's no drugs actually removed from the market due to the torsade or pluralismic risk. But the problem here is we have a hERG inhibition compound collection, we have a QT prolongation compound collection. This all represents a large collection of the compounds or the drug candidates. But the actual compounds that has the potential pluralismic or torsade risk only represents a very small subset of these compound collections. So this leads to, the strategy is not very specific and leads to a lot of unnecessary early compound titration during our drug discovery and development process. So this leads to the CiPA paradigm or the CiPA initiative. So now we're looking to, there's multiple cardiac ion channels. We know that QT prolongation doesn't equal to pluralismic risk or torsade risk. hERG inhibition doesn't equal to pluralismia or torsade risk. The good evidence or example is that Prozac or verapamil blocks hERG, but also inhibit the CAP1 to the L-type currents at roughly the same concentration, and they are cardiac safe. And then there's other examples of these compounds too. So the basic idea is that you have a fine balance of inhibition between the inward currents and the outward currents actually that cancels out the effects of each other. So our hERG-centric or hERG-only focus has a very negative impact on the drug development. As I just told you, it causes unnecessary early compound titration and causes a lot of potential of losing good drugs. So thus we have this new paradigm of cardiac safety evaluation, which we call CiPA, Comprehensive In Vitro Arrhythmia Assay, which basically contains 3 different approaches. One is that we test the effects of the test compounds on multiple cardiac ion channels, and then we feed all this data into a computerized in silico model reconstruction to predict the risk of pluralismia or torsade. And then we test the compounds in the stem cell-derived cardiomyocytes model to confirm this finding. Actually, another aspect of this is then we confirm the finding in the early clinical trial data. So what are the candidates for the current? Like I just told you, for the current candidate, we want to look into the cardiac ion channels that has that can balance out the outward current inhibition and inward current inhibition. So we have hERG-centric strategy, which has the hERG blockage, and now we're discussing about the possibility of a sodium fast current and sodium late currents test in the L-type CAP1-2, and also the candidates. We also have Kiwi long QG, Kiwi 7.1 link channel, and KIR21 a rectifier channel. But we are still in discussion about what's the final list of the channels that we're going to test include into this cardiac current candidates. For this particular study, I'm going to just show you validation data on 3 different channels: the sodium 1,5, both the peak current and late currents, the hERG channel, and as well as the CAP1-2 channel on the IonFlux. So I want to mention that actually here at the Eurofins Ion Channel Service Team, we offer a monthly service that we test 8 different cardiac ion channels on the IonWorks Quattro and menu patch clamp to evaluate the multi-channel effects on the cardiac ion channels. And we are currently also validating these currents or channels on multiple ion channel platforms, which include the IonFlux platform I'm going to show you in just a little bit. Here I want to show you a video introduce you the platform. So IonFlux HT platform was a plate-based microfluidic design that the plate was defined or divided into 32 different recording patterns. In each recording patterns, you can see that we have 8 compound wells, 2 compound wells for internal solution, and 1 well for cell. And then the plate was inserted into the platform, and everything was controlled by the pressure. There's no moving parts. The cell addition or the cell flow the introduction of internal solutions and introduction of your test compounds are all controlled by the pressure to ensure the fast introduction and removal of all the components, particularly test compounds. So the cells start flowing driven by the pressure, and then we apply a negative pressure on the trap, which are the top two panels that you can see on this video, and then to attract the cells into the traps. And then after that, after the seal was obtained, you can see that we can apply another bigger pressure to obtain the whole cell recording. And then you start perfusing GABA on the different recording traps. And you can see that the traces show you a dose-dependent response of the GABA. I've noticed that on your computer screen there might be a little bit lag on the video, which actually later on you're going to have the PDF version of the slide deck, and you can also watch this video on the YouTube. So like I just told you, the IonFlux HT system is a microfluidic plate-based design, so it gives you a lot of flexibility in the assay development and design. And as well as a very fast, super fast actually compound introduction and removal within 100 milliseconds. Also temperature control, and as well as the individual cell trap recordings that you can test multiple targets on the same plate. First, we validated the Hack-Herc channel on the IonFlux HT. In this set of experiment, we look into different aspects of the experiment. First is the current stability. We test the time-matched vehicle control throughout the time course of the recording to make sure that the current rundown is minimal, because as you know, the current rundown might be a problem for your potency evaluation because the reduction of your signal, even without introduction of the compound, is a problem. You can't do a rundown correction, whether it's linear or exponential, but sometimes we know that just by adding the compound, it causes a unpredicted drop of the current. So we want to make sure that our current recording is very stable across the time period we're applying the compound. And then we test a couple of reference compound pharmacology on the cells to make sure that they agree with the historical data in-house and the published literature data. And then we test the intra-run variability on different days on the same reference compound to make sure that our assay is robust enough. And all this data was generated on the Eurofins Precision Hack-Herc cell lines. And we also did a cross-platform correlation, compare the data collected from the IonFlux to the Q-patch. On this slide, you can see we use a pretty standard Herc voltage protocol. And the upper panel, you see the current stability. The current throughout the recording time period is pretty stable. I show you 3 representative lines. And on the bottom panel, you see the CiPA price. The baseline recording is very stable. And then after that, you have a very nice step-like dose-dependent response of CiPA by adding higher and higher concentration of CiPA. And towards the end, you have a nice washout because the solution is a continuous perfusion, and solution exchange rates on this platform is really fast. And then we test a couple of reference compounds on the Herc channel. We test estamazole, cisapride, E4031, tafenidine, and as well as the time-matched vehicle control. And you can see that all the reference compounds, the potency data, it's really close to reported many patch clamp data, and the vehicle control shows minimal rundown across the time period of the reporting. In the next set of experiment, we look into the intraday precision to test the robustness of the assay. We pick 3 compounds, cisapride, estamazole, tafenidine, as well as time-matched vehicle control, and test those on 2 different days. You can see between day 1 and day 2, the IC50 value is really close to each other, show that our assay on the IonFlux, the Herc assay on the IonFlux is very robust. Then we compare the potency value of the reference compound between QPatch and IonFlux. You can see that they both agree with each other, and the time-matched vehicle control looks very good with minimal rundown, current rundown. Next couple of slides, I'm going to show you some sodium 1,5 data on the IonFlux. Again, we look into the current stability with time-matched vehicle control. We look into some reference pharmacology of the reference compounds. And we also look into the late current pharmacology induced by the ATX2 and blocked by ranolazine. And again, all the data was generated with the Eurofins precision cell line NAP15 in the HEC background. In this slide, you can see I show you use a two-course protocol to test the state dependency of the test compounds. In Rock's presentation, he already explained that the state dependency of the blockage, so I'm not going to go into the details of that. But you can see, again, our vehicle control shows a very stable response across the time of the recording, and then you have a very nice step response of lidocaine as you apply higher concentration of lidocaine, and then until fully blockage of top concentration, and after that you have a full reversal or washout of the lidocaine blockage effects and bring the current back down to the baseline level. And you can see that the lidocaine shows the post-1 IC50 of 737.9, close to 1 millimolar, and the second pulse shows IC50 of 82.7, shows a very state-dependent inhibition of lidocaine, which also, again, this data agrees with the literature values. Then we do the same thing across all the human NAV receptors we have in-house. We have NAV11 through 18, but here the only difference is that we actually use a usage-dependent protocol. We use a 20-pulse protocol to test the usage or frequency dependency of the test compounds. We use either tetracaine, lidocaine, or ABA compound for the NAV18. So NAV11, you see there's a usage dependency between pulse 1 and pulse 2, pulse 20 for the potency data, and similar findings on the NAV12. And on the lower panel, you can see vehicle control, time-matched vehicle control of each receptor or channel, and show you minimal current rundown throughout the recording. Here's the data for 1.3 and 1.4. Again, the potency value agree with the published literature value as well as our in-house data. 1.5 and 1.6, and 1.7 and 1.8. On NAV18, I want to draw your attention to this ABA compounds. It's very difficult to get the potency value down to low nanomolar range on the automated patch platforms for this ABA compounds on the NAV18. But in here, I showed you that actually due to the fast solution exchange and continuous perfusion feature of the IonFlux, we can actually get the potency value down to the low nanomolar range. And here you see the vehicle control data. The baseline response is very stable across the time. So next couple of slides, I want to show you NAV15 late current recording. First, what we do is, again, on the top panel, this is the baseline response, and then we add different concentrations of ATX2, and you can see there's a nice dose-dependent response. And then on the lower left panel, you see that's the vehicle controlled with little to no late currents, and then on the right panel, you see there's a dose-dependent response, ATX2-induced late currents. And we compare the IonFlux potency value to the QPatch, and they are very close to each other, and as well as agree with the historical values and published literature values. And we also use the temperature control feature of the IonFlux system, test the ATX2 compounds at 37 degrees and room temperature, and we didn't see there's a potency shift between these two different temperatures. We have another cell line with beta-1 auxiliary units, and we want to test that. So again, we do the similar experiments, induce ATX2 to a dose response, and you can see the baseline response, again, it's very stable. And then if you introduce a different concentration of ATX2, it shows a very nice step-like dose-dependent response. And again, here's the vehicle. Here's the dose-dependent induced late currents by the ATX2. Similar comparison, we do 37 degree on the IonFlux room temperature and compare that with the Q-patch. There's a shift between Q-patch potency values and IonFlux, but we don't see a temperature-dependent shift on the potency of ATX2 on the NaV1.5 beta-1 cell line. And next, I want to show you that we can block this ATX2-induced late currents by ranolazine, which is a known late current blocker. Again, you see here's the baseline response. And then we use ATX2 to induce the late currents, and we use different concentrations of ranolazine to block it. You can see there's a nice dose-dependent blockage of the late currents with washout reversal. And we compare the potency value again between the IonFlux and the Q-patch, and the potency value is really close across different platforms. So in the next set of experiments, I want to show you some preliminary data we have on the CAP12 cell line. The CAP12 cell line was known technical challenge or difficult on the high throughput automated patch clamp platforms, mostly due to the current stability or current rundown. Here I want to show you with IonFlux that we can get a very stable baseline response of the time edge vehicle control. Historically, with the way we're dealing with the current rundown of CAP12 is actually on the Quattro we use perforated patch, and Quattro is probably shows the best or with a minimal current rundown compared to other true wholesale current recordings such as Patch Express, Q-patch, or other wholesale automated patch clamp systems. Actually, right now, when people are dealing with rundown problems of CAP12, even on the Q-patch or Patch Express or other wholesale patch clamp systems, we actually also try perforated patch instead of wholesale recording. But here, I want to show you some very exciting preliminary data actually on single hole recording modes, which is the upper panel, and population modes, which is the lower panel, and the superimposed calcium currents over different time, that they are pretty stable throughout the reporting time period, which is about 10 to 15 minutes, with minimal current rundown. Actually, this reporting was achieved by wholesale reporting. This is very exciting, and we're continuing to test some pharmacology of the reference pharmacology of CAP12. On this slide, I want to show you the capabilities of the Anchannel service team here at Eurofins Panlabs. We have a couple of menu patch clamp rig. We have Q-patch system here. We have several IonWorks Quattro system, as well as the IonFlux HT system I just showed you. We also have a high content image express system, as well as the Axion Mastro system, which we currently use for carbon monoxides and EA recording. We also have a couple of plate readers for fluorescent signal readings, and as well as lots of liquid handlers for lab automation. With that, I want to thank you for your time and audience, and thank you very much. If you have questions, you can ask now. Thank you.